Thank you. It is a great pleasure to be here today and have this opportunity to tell you a little bit more about what we do in Exeter in the UK with exome sequencing, mainly for children with very rare genetic disorders and a small number of adult patients as well. I'm going to tell you a little bit about who we are, what we do, and then the key message from my talk of today, I think I'll just deliver that now to you. The thing is, exome sequencing is extremely flexible. There are so many strategies out there that you can use. You can use panels and use a phenotype-driven strategy. You can look only at parents. You can look at affected children from consanguineous unions. And you can also look at trios. So there's all this flexibility out there. But the message is, if cost is not an issue, if you have a patient with a non-specific phenotype, then the best strategy for those patients is to use a trio analysis, where you don't have a pre-hypothesis of what is the causative gene, and where you just go for a gene-agnostic approach. So that's the message that I want to give today, and hopefully I'll convince you with a few examples of when a gene panel analysis didn't make it. We are NHS National Health Service hospital. We provide diagnosis for patients that are referred through clinical genetics. We do have a small proportion of patients that are referred by pediatrics directly, bypassing clinical genetics consultation, and a small number of patients that are referred from neurology as well. But the bulk of our work is patients that are referred from clinical genetics. We have close links with University of Exeter. We share a research building with a medical school. And of course, the big strength of being a university hospital is that we have research colleagues that develop software tools, ideas and projects that can be very quickly and easily translated into clinical practice to benefit patients quickly. We are a research-intensive centre in Exeter, in the southwest of England, very famous for working in diabetes, and we do exomes and genome sequencing for diabetes, looking for novel genes. And a lot of the knowledge that we gain on our work on diabetes technology, we are transferring to patients with rare genetic disease. Until November 2018, we were using a different provider to twist, and we were struggling with our copy number variant calls, and we were also struggling with the fact that we needed 3,000 nanograms of DNA to do exome analysis. A proportion of our patients are actually deceased. There are termination of pregnancies, fetal samples, for which the amount of DNA is very small, or sometimes it is sufficient, but the sample is precious. So we are a bit reluctant to use a precious sample, use it all in an exome, and then one day if we want to do a genome, we won't be able to do it because we've used all the sample. So there was this urgent need to have a protocol that used little DNA, and that gives us uniformity of coverage to empower our ability to detect copy number variants. So being a diagnostic laboratory, we are UCAS accredited laboratory. We need to justify, validate every single change that we make to our protocols. So we just did an extensive comparison of our previous kit with the twist, and we saw an improvement in the coverage that we were getting, an improvement in the uniformity and the performance of the assay, and we also saw that we would need less sequencing to achieve the metrics that we had set up for these patients. So we were satisfied that we could make this change from our previous vendor to twist. And we also run the genome in a bottle sample from the HapMap project, and we looked at our sensitivity to detect single nucleotide variants. You see this is the sensitivity with our previous vendor, and you see a small increase on our sensitivity for single nucleotide variants. And what we found was actually a much welcome improvement in our ability to detect indels. So that was very good, and at this point we were happy to start using the twist. We are not a high throughput laboratory. We are one of the few centers in the UK that offers exome sequencing nationally, but cases are selected carefully, and we've not done masses. We've done in total over 1,000 families. So this is just a summary of the metrics. I think for us, the big thing was be able to use less DNA for these patients. I think, like everybody else, we struggle with the copy number variants. We've tried several different tools, and I just wanted to put out there that one of our bioinformaticians developed this SavvyCNV tool that we find is also the combination of the twist and the SavvyCNV really improved our ability to detect copy number variants. This is available on GitHub if anybody is interested. So I'll dive in a little bit in more of detail on what do we do for our patients. So we started doing exomes in 2013, and we favor this gene-agnostic, inheritance-based approach. So what we do is we use a pipeline based on GATK, pretty much like most of the standard things that most of you will do for exomes. And then we don't have commercial tools to visualize the data, but so we just look at an Excel spreadsheet where for a trio analysis, you'll have the list of compound heterozygous variants that are rare. So you will only have 2 to 4 compound heterozygous variants. You then have the list of the homozygous variants, again, 2 to 4. If the proband is male, we will look at rare X-linked variants, and we then have the list of the de novo variants. For a trio, we'll have anything between 8 and 12 de novo variants. So what the scientist then does when they look at that trio is they ask the question, can I see a variant in a gene that fits the phenotype in this patient? So we are looking for an answer to the clinical question. We do not report incidental findings. That's our policy, and it's very clear to the clinicians who refer to our service that that's what we are doing. And our bioinformaticians also were able to incorporate as we went along, we saw the need to have a UPD section so that we could capture those. And we also have a section on imprinted genes that we look at. And we are doing exomes and targeting the exons, but of course mitochondria are very abundant. So you can find sequencing data for the mitochondria in the off-target reads. So our bioinformaticians developed a tool that allows us to look at those untargeted reads and look for mitochondrial variants as well. So a trio enables us to look at all those different mechanisms, genetic mechanisms. So it's our favorite. But of course, there's cost, and it's cheaper to do 1 sample than to do 3. Actually, it's not. It's cheaper to sequence 1 sample, but the cost of analysis for a singleton proband-only gene panel is actually quite high. My job is to look at the variants, so I'm not a bioinformatician, I'm a biologist, and I do variant interpretation. I spend 20 minutes looking at a trio. Sometimes I find the variant in 5 minutes, but I spend 20 minutes looking at the genes. But if you give me a gene panel, I will spend the whole morning looking at it. So yes, it's cheaper because you're sequencing only 1 sample, but the cost of analysis is not lower. We also can do, so when we do a singleton, we can look at a panel of genes, but we can also do other sophisticated things. For example, if you have a family where the segregation pattern suggests that you might have an X-linked disorder, then instead of applying a panel of genes, we can apply a genetic approach where I will say, I want to see all the rare variants that are on the X chromosome and see if I can find a diagnosis for this family. For consanguineous families, we can look at shared homozygous variants between affected siblings. We actually found that it's easier for these consanguineous families in our hands, it's easier to just sequence one of the children and just look at the homozygous variants in that child. And then we do a Sanger study to see if the variant co-segregates. One of the things that we developed a few years ago was for these families where there was no DNA on the fetuses and there was only DNA on the parents. It's very simple. You have a hypothesis. This is most likely to be a recessive disorder. If you just sequence the parents and you develop a pipeline that will look for rare variants in shared genes, homozygous or compound heterozygous, can you find the cause? And of course, this is very important for these families because the recurrence risk for recessive conditions is 25%. And most of the patients that we are testing, they are not only after a diagnosis for their child. The greatest proportion of the families that we test want another pregnancy. So they are after a diagnosis to use for prenatal testing. So that's our pipeline, pretty much standard, and we use GATK. Exome depth, this is an exome depth we don't use anymore. We now use SavvyCNV, and we use Alamut and the usual databases that most people use that are out there. So this is some of our results for when we looked at only the parents. So we do a parental analysis looking for recessive conditions, because of course the a priori probability of finding a diagnosis is high. We have a quite good diagnostic yield of 52% in these patients. And if they have at least 2 affected pregnancies, then the diagnostic yield is at around 62%. And that's just a reflection of the biology, really. So I'll show you, here I have more data. So I have done around 80 families where we sequenced only the parents. And for those 80, the diagnostic yield is now 60%. And then, of course, because we now have the twist exomes that allows us to use less DNA, we actually are no longer encouraging people to do the parental analysis, and we prefer to go for a trio because, of course, there's always the possibility of parental mosaicism, and if we do a trio, that allows us to find parental mosaicism. When we do trios, we've done nearly 1,000 of those, we find a diagnosis in 37% of the cases, and a likely diagnosis in a further 1%. A likely diagnosis is a very good biological candidate to explain that phenotype. For example, let's say the child has brain anomalies. The gene is expressed in brain. There's a mouse model that recapitulates the human phenotype. However, they're not families reported. Because we are doing diagnostic, we cannot issue a diagnostic report for those cases. So what we do with those cases is we try to find people out there that can do research and collaborations to allow us to issue that diagnostic report for families to use prenatally if they wish so. And of course, that is one of the advantages of using a trio as well over a gene panel approach. When you use a trio, because you're seeing everything that is rare, you will see something that you might want to follow up. Whereas if you have done a singleton panel analysis, you do not leave space for novel discovery. And when we do a panel analysis, we have a very good diagnostic yield, 39%. I think it's because for the patients that we are doing panel analysis, they have a very specific phenotype. They are very well characterized from the clinical point of view, and the clinicians have a very good hypothesis of what is the culprit. So we do find a diagnosis in 39%. We have a little bit more of uncertainty in this group of patients, so we find 3% of likely diagnosis, and we pretty much get stuck with those because in most of these cases we did a panel rather than a trio because one or both parental samples were missing. So we cannot determine if the variants are de novo or not, and we are left with these good candidates, but we can't go further. Just to show you how our diagnostic yield from when we started. When we started, we told our favorite clinicians, we want to try do exomes. Can you give us your best cases? And they did. And of course, our diagnostic yield was 100%. Once you start with 100%, it can only go one way. And what we are finding is pretty much what people have been reporting in this meeting and others. The diagnostic yield seems to be around 40% for exome sequencing in patients with rare genetic disorders. So we've now got over 1,100 families, found the diagnosis in 420. Most are SNVs and indels, 5% are CNVs. I think this is also because most of our patients have had an array CGH, had deletion, causative deletions and duplications reported that way. And we also see a very small proportion of our diagnoses are mitochondrial variants in printing disorders. We have a very small proportion as well of children that have two separate diagnoses as well. So this, we have 312 different genes, just to show that most of the variants and genes are unique to these families that we tested in our cohort, but they are of course known disease genes. And I have a gene cloud of the top culprits. So our top culprit is RYR1, and that's not surprising because it's a huge gene that causes a congenital myopathy. I think we have 7 families with variants, pathogenic variants in RYR1. Another big hit is ATAD3A, which is a neurological phenotype, and I think this is a big hit because it's a gene that was only discovered in 2016. There's a small number of families reported out there, so the more people we start doing exomes on, the more we will find that have these novel genes that seem rare, but once you know they are out there, you'll find more and more. Some genes are quite common. For example, Soto's syndrome is quite common, is a very recognizable syndrome. But then, of course, what we are finding in these exomes is patients that are atypical. So they have a known disorder, but the clinician would not have guessed, based on the set of this morphology, would not guess that that was the gene. So we see lots of very well-known disease genes, but they are just atypical presentations in these patients. And others are because there are fetuses that were terminated, so most of the literature describes postnatal cases, and of course the antenatal phenotypes are not as well described. I want to say something about panels, take the opportunity that we have lots of you here. In the UK, there is this big drive to use PanelApp to design virtual panels. And what it is, it's like a database of over 300 panels for different phenotypes. Now, the beauty of this is that anybody can sign in to be a reviewer for PanelApp. And if you have your favorite gene, can you just go on PanelApp, have a look if your gene is in the panel where it should be, and to help us all, because this resource is available to all, to help us all be better at designing panels. Because, of course, our preference is to do trios, but panels also have lots of strengths. So if we can get the panels right, that will be extremely helpful for everybody. I will give you an example of 3 recent diagnoses that we made in the last few months, where a panel approach would have failed us. So this was the case of termination of a pregnancy. And when the referral card came through, the only thing it said was polycystic kidneys. So we did a trio analysis, and we found biallelic variants, clearly pathogenic variants, in this ETFDH gene. And of course, this has a huge impact for the family because it means that the recurrence risk for next pregnancy is 1 in 4. So that was the answer that they were after. So when I communicated this result to the clinician said, but I have done a panel for polycystic kidney, and ETFDH is a known gene to cause polycystic kidneys antenatally. So the gene was not in the panel, so it was, poorly designed. Somebody didn't notice that it was there, but it should have been there. But it was in a metabolic panel. So if that hadn't been a fetal loss, so if it had been polycystic kidney plus the typical metabolic presentation, then we would have picked up. So we now communicated to the lab who provided that testing and asked them to make sure that that gene makes it to the panel. But it is a shame, isn't it, that people spend £1,000 to do a panel and you'll never find a diagnosis because the gene is not there. So this was another case where we had a bit of a discussion in the team because our typical presentation is congenital malformations or developmental delay. And this was a 4-month-old baby who had anemia and other hematological disorders. So my colleague came to me and said, "Oh, Julie, I'm not sure about this one. Anemia? Are we going to be able to interpret the result and find, this is not what our expertise." And I said, "I think we should give it a go. And then we can ask for help." So that's another thing that we do a lot because we are doing these rare diseases. We find genes in disorders that we've never heard of. So what we do is we phone a friend and ask people that have the expertise in that disorder to help us interpret the variants that we find. But this was very satisfying. What we found was, again, a recessive condition. This baby had biallelic truncating variants, clearly pathogenic, in the SLC46A1 gene. So this is a folate malabsorption gene. And by finding this diagnosis in a baby that is 4 months old, we were giving them the possibility of having treatment, just supplementation with folate, and of course that will avoid neurological damage, and this child will not have the classical presentation that you would see with patients with mutations in this gene. So if you see a patient that is 5 or 6 or 7 years old, they will have severe intellectual disability in addition to the hematological anomalies. So we actually were able to stop this child becoming neurologically damaged. So this is an example of where applying a panel we would miss that diagnosis because the only phenotype that we had was anemia and hematological disorders, and that gene is not in a hematological disorders panel. That gene is in an intellectual disability panel, which we would not have applied here. So again, another example where we would miss a diagnosis that would be life-changing for this family. So I've said that. So I went and added the gene to the right panel, and we will not miss that one. The last case is another example of when a gene would be, a diagnosis would be missed by applying a panel just because the gene is too new. It's too novel. So you have to design panels at a certain point. You can't be designing them every day or every week. But genes are being published every day and every week. So your panels, when you apply a panel, by definition, you are using an outdated method. So this was a child that was identified on ultrasound scan, I think was on the 20 weeks scan, as having an ambiguous genitalia and a diaphragmatic hernia. And when the clinician contacted me about this one, again, I was not sure because this kind of phenotype seems a bit chromosomal. So I was thinking, okay, we can try, but I think the chances of finding something are a bit low. And what they had was a de novo truncating variant again in a gene that is described in 5 or 6 families, all of them presenting with ambiguous genitalia, diaphragmatic hernia, and cardiac defects, which this patient didn't have. And the gene was only reported last year, and it was not in any of the panels. So we've added that to the panels now. So I hope I convinced you with these cases that a trio analysis is a powerful approach for patients with non-specific rare diseases. And of course, it allows for reanalysis of the data at a later stage quickly as the individual grows and novel features become apparent. You can then revisit the data. And of course, it allows you to think about these atypical patients that don't fit the clinical descriptions that are out there in the literature. And of course, when you do this trio and you make any improvements in your pipeline, you just need to run the data again rather than sequence the patients from the beginning. And of course, when you do a trio gene agnostic analysis, allows the scientists to see very interesting variants that require follow-up. Okay, so as I said, we are a diagnostic laboratory, but when we see things that warrant further research, we tend to use Gene Matcher, and we put the details of our gene out there. And in the last couple of years, we found all of these genes as interesting biological candidates. So if you spot any of there that you have families for, please let us know and see if we can, if we have a match on the phenotype side of things. And between last year and this year, we've published 4 novel genes thanks to the trio approach that allows us to have this, to think about genes that are interesting. So it's been very positive. So yes, the takeaway points is that exome sequencing is highly flexible. There's a high success rate. Of course, the high success rate is also because we introduce biases by selecting the best patients, the most likely patients to have genetic etiology. Of course, there is a high potential for uncertainty. I mean, there are lots of laboratories with whom, people with whom I talk, that explain that they prefer to do panels because you won't have surprises. So yes, there's a high potential for uncertainty, and that's an important consideration. But if you ask for help when you find uncertainty and join efforts in unsolving the uncertainty, I think that's the best thing for our patients. We've been extremely lucky in getting support from the wider genomics community, both to find novel genes, but also to interpret the variants that we find that are tricky. So yes, I just will say again that the gene agnostic trio analysis is really, really powerful. So what is the future for us? I don't know if you've heard, but the NHS is pushing with the genomics agenda. Genomics will be available to everybody, free at the point of care. And Exeter is going to be one of, we don't know one of how many, there was a talk of one of 2 or maybe the only one nationally providing rapid exomes. So our future is about doing more and doing it faster. So, easy. Our turnaround time for these rapid exomes has been set at 21 days. So we will have children that are in neonatal intensive care units, NICUs, PICUs. We'll have pregnant patients, and we will be looking at doing exomes and providing diagnosis for those families in 21 days. We have been able to do exomes in 8 days. That's the fastest that we have been able to do. Everything has to be, the wind has to be in the right direction for us to be able to do that. So the challenges for us is to make it sustainable, make it bigger, and make more, do better for our patients. So it's exciting times. And of course, the work that I presented is the work of a lot of people, genetic technologists, scientists, bioinformaticians. They are key to everything that we do. And of course, the patients and their families. Thank you.